Ma'am, I am not able to hear your voice in today's presentation meeting.
electrophoresis, and based on the size or the molecular weight, or in this case for DNA, only size will make a difference. Based on the size of the fragment that is generated, they will get resolved along this polyacrylamide gel. So the shorter a fragment, the longer it is able to migrate. Just a second. I think my, yeah, maybe my audio is okay now. Can you hear me? Yes, ma'am.
Ma'am,
Yes,
now
ma'am. We
we
can.
can hear you, but from half 1.5 hour, you are not hearable to on meeting.
Oh, they should have interrupted me. I've been speaking for like an hour. Oh my
Yes,
God.
ma'am. I'm interrupted you, but maybe yeah,
Okay. Oh, okay.
I mean,
Yeah, I'll record the audio separately and then yeah, it kept showing me there's an issue with my speaker or something. I don't know what was wrong because it was connected. Uh, Never mind. I'll upload it again with a separate audio if necessary. Uh, you can go through the slides. I think it is informative enough, but okay,
ma'am.
that's yeah. Anyway, if there's any doubt, you can just let. But yeah, I'm so sorry. I didn't realize. I think I said once in between to also let me know over chat. Did anybody write something? Okay, never mind. It has been too long. We'll figure out a solution to it. Don't worry. But anyway, so I was talking about Sanger sequencing. So yeah. So I'll end it here only till now. Moving forward, this was the traditional method of Sanger sequencing, and it involved these four reactions, and then subjecting them to polyacrylamide gel electrophoresis for separation of these fragments, and then to dictate what is the readout or what is the sequence that can be concluded from these reactions. Now, while we keep calling these first generation techniques primitive, it is quite surprising that there still is so much relevance of Sanger sequencing techniques even in today's time. Whenever you clone a gene of interest into any plasmid by using restriction digestion, or you carry out some sort of a site directed mutagenesis in your lab, or you PCR purify some region and clone it into a different vector, any cloning method if you perform, then you have to isolate the bacterial colony's DNA and then subject it to Sanger sequencing to confirm that yes, the sequence that you expect is still there. So in today's time, even despite all of these advanced techniques that have come into play, Sanger still stays relevant. The only point of difference at this currently is that the type of tag which is used has been changed, and the process has been subjected to a lot more. Um, it is a lot more affordable and a lot more easily accessible in today's time. So, like I mentioned, Sanger method was it involved the use of chain elongation termination. This I'm discussing the traditional method. So it involved chain elongation termination, which is using DNA sequencing with the help of polymerase and some special nucleotides. What do we mean by special nucleotides? They are basically DD-NTPs, which are dideoxynucleotide triphosphates. So what happens when you incorporate okay, let's put it this way. In each reaction mixture, you will have your template DNA, that is this DNA sample. You'll have your primers of interest. You'll have your polymerase enzyme, which is capable of carrying out Okay, hi. I'm so sorry this class has been just there's been way too much disturbance in this class. If there's an issue, don't worry. We'll discuss this again in the next class. It's not a problem. At least the key topics, because I don't know what is going on. My internet is terrible. Okay, so quick recap. The first method that I talked about. I hope the screen. Okay, no, I think I need to share screen. Yeah, should be okay now. So the first method that we talked about was the Maxim-Gilbert method, which involved chemical degradation, and the degradation was carried out by certain chemicals that stopped or that broke DNA at different positions, and these positions were dictated by the type of chemical that was used. So, based on which base is present at a particular position, a chemical was able to act, and that dictated how that chain will get terminated. And based on that termination, you could not termination, degradation, you could find out what is the base that is present in a particular position within the sequence that you are trying to analyze. In contrast, the Sanger method, which is something that I've already discussed, is relevant in extremely relevant in today's time as well, involved the chain termination method. And when we say chain termination, how is it different from the previous technique? The previous technique talked about DNA getting broken or interrupted, or rather, broken is the correct word, because there was a particular chemical treatment which was carried out. So that was degradation. In this case, DNA is not broken, it is simply the end of a polymerization reaction, because there is no more, it is not possible to incorporate any other nucleotide bases. So the chain gets terminated at that particular site. So, taking this into consideration, the chain termination method involves the use of these dideoxynucleotide triphosphates or DDNTPs, which are also monomers of DNA chain. The difference between two is the lack of a hydroxyl group at the sugar's third carbon, as a result of which the next nucleotide base cannot be incorporated into the currently elongating chain. How that actually affects everything is that, say, for example, this is the initial steps are the same. Like there's a DNA sample which gets denatured, and then it undergoes amplification. And after amplification, when the Sanger sequencing reaction actually takes place, then instead of having those chemical reagents that we have discussed for Maxim Gilbert, there is the incorporation of one of these individual DDNTPs along with the presence of all four DNTPs. DNTPs are necessary so that normal polymerization can take place. Individual DDNTP, shown here in different colors, is necessary to ensure that termination only takes place at one particular base for each of these polymerization reactions. Now, as a consequence of this, wherever, for example, in this reaction, wherever there's a G, termination will take place here for A, here for C, and here for T. As a consequence, when each of these four reactions are then run onto a gel, you will be able to figure out what is the termination base at that particular position. Um, okay. Yeah, we are just getting interrupted multiple times today. Uh, anyway, so currently the advancement of technology has led to a situation where the same Sanger method is now performed with fluorescently labeled DDNTPs instead of them being radioactively labeled as was previously carried out, and then following it up with capillary electrophoresis. So while it was necessary to set up 4 different reactions for each base with the old method, like I just mentioned. Currently, what you can do, you can minimize the infrastructure that is necessary for each Sanger reaction and instead carry it out in a single tube where each of them will carry 4 separate tags, fluorescent tags. And then this fluorescent tags can be detected using lasers where each of them will fluoresce differently, and then you can decode what is the position of each base within the nucleotide sequence that you are using as your input sample. So while the initial steps are the same, within the reaction mixture, your DD entities become labeled and they are all present within the same reaction tube, which is shown here within the PCR mix. So the fluorescently labeled DNA sample, which will now get sequenced, will have all these possible combinations where at each position, when this labeled DD entity gets incorporated, then at each site you can eventually identify what is the base that is present. And once capillary gel electrophoresis is carried out, the DNA sample passes through your uh, or when laser is able to sort of when the laser beam falls on the base which is present at that position and the emitted fluorescence is detected by the detector, you get a readout like this. So this is overall how it works, and what you get, this output that you get is called as a chromatogram. And the sequence chromatogram is extremely crucial because the presence of a sharp peak, like you can see here, you get even sharper peaks. It's just a schematic representation, but this sharp peak is extremely important to make sure that at a particular site only one base specific or dye specific fluorescence is emitted. There is no overlap, because a bad sequencing report will have something like a broad peak, like a blue and an orange overlapping at the same site, or it can have like sort of a broken peak like this, which are often also called as shoulders instead of there being sharp peaks. And those are all going to give you improper or inconclusive or slightly less certain results. So those are not very trustworthy and therefore should not be that is not a report you should accept from a company who you're getting your sequencing reports from. So they should have clean peaks which will dictate that this is the base that is present at this site. Now over time, as more and more as more and more technological advancement has taken place, it has made this technique extremely affordable. In today's time, you can get Sanger sequencing done from uh, as little as I think 300 or 400 rupees for one reaction. It's very cheap in today's time. So um, yeah. So after capillary gel electrophoresis, DNA fragments get sorted based on their size difference, like we have previously mentioned. The fluorescent dye is then detected, and it occurs through spectral detectors called as charged coupled devices or CCD. Then softwares, many available. There are softwares like Chromas or any freely available online software can then be used to interpret and analyze these sequences. Next, moving on to second generation sequencing. So please keep in mind that right from this point onwards, from first generation sequencing onwards, things progressed a lot faster because because everybody understood the relevance of using such high throughput techniques. So a lot more companies made it a lot more affordable. However, there were some shortcomings with second generation techniques as well, which is why the third generation techniques have come in, which have made techniques A, less time consuming, B, very easily accessible, and C, a lot more affordable. So compare anybody any lab that carried out these genomic techniques 15 years ago versus not even 15, 10 years ago versus now, and you'll see the difference. You'll see the cost effectiveness of the process and how much more people are recognizing its relevance in today's time. Now, coming to second generation sequencing, the first major difference that occurred or that was taken into consideration was that you are trying to sequence the entire genome. In contrast to Sanger, here you are focusing on a high throughput method and you want the entire genomes to be sequenced. This is this falls under the purview of NGS or next generation sequencing. And for this, there were several kits and equipments and many more possibilities, which are designed to make it cost effective and much more time consuming. And here, Okay, so there are multiple platforms also that carried out this second generation sequencing. Um, but I am majorly focusing on only the Illumina based method. It is most acceptable in today's time. So Illumina is a company, a sequencing company. You might already be familiar with it. It acquired this analyzer and commercialized it. And HiSeq, MySeq particular platforms in particular are something that a lot of labs actually have. It's present in our facility here also at IIT Kanpur. So these are smaller sequencing platforms which people are able to place in their own labs if they want to carry out sequencing on a small scale within their lab. But large scale platforms, of course, are taken care of by companies which are able to make it a lot more cost effective because obviously they are um, able to perform sequencing reactions in thousands in one day. And they also have the high like those extremely efficient high throughput processing system which are able to analyze a lot of data in a very minimal amount of time. So commercialization of these techniques is the common way to go. Now, here, just to understand overall what has changed in the sequencing, um, in the domain of sequencing. So, the amplified template DNA for sequencing, which is similar to what we have already seen for Sanger as well, it creates these miniature colonies called as polonies by bridging PCR. In order to, again, let's take it step by step. In order to sequence data, what Illumina does is it uses reversible dyes. These dyes, they are added to DNA strands and these colors make it possible to recognize nucleotides, much similar to what we know about Sanger in today's time also. The blocking group and fluorescence with a distinct color for each nucleotide are revealed when a nucleotide binds, when it is incorporated into the currently elongating sequence or currently elongating DNA sequence, then that gets detected by a laser or that gets detected by a fluorescence detector. To create DNA fragment clusters, the adapter segments are joined to single strands and they are put inside the flow cell. Then bridge amplification is carried out, and the four types of nucleotides they get introduced, and each actually I'll show you a video. I don't have it today. I'll show it to you in the next class. That is going to clear it in a lot more detail. But basically, what happens is you take cells, you isolate all the whatever material you're trying to sequence. In this case, we are talking about DNA sequencing. So all the DNA is isolated from the cell. It is subject to fragmentation because it's not possible to sequence very long stretches of DNA. So we have to fragment it. Now, that fragmentation can be done in many methods. Usually, it involves ultrasonic fragmentation. Sometimes, certain like equipments, which, for example, are called bioruptors or Covaris, for example, it uses Covaris also, for example, is able to, you can set like a range within which DNA fragmentation will be carried out. It creates pulses of sound waves, which is able to generate breaks within the genome and generate fragments within a certain range, like for example, 700 base pairs, 500 base pairs, so on and so forth. Now, after that DNA fragmentation is carried out, this DNA also keep in mind is already ligated to adapters on either end. This adapter A and B shown in blue and red. This is extremely important because the adapter is what will bind to the flow cell um, to the area where sequencing will actually take place. So this fragmented DNA is then bound to adapter and that creates a library. A library in this case will depend upon what sequencing kit you are using, what is the instrument you are using. So these are all intellectual properties of the companies which are concerned. So after library preparation, what is allowed to happen is that the library is allowed to hybridize or bind to a flow cell. A flow cell is just a small portion of the plate on which sequencing is being carried out. So after the library has hybridized there, then what you do, you perform bridge amplification. Now this bridge amplification step is something I'll show to you in a video, but for now, just think of it as a specialized way of performing PCR, polymerase chain reaction, which allows for more and more amplification of these sequences. Because right now, within a cell, you'll only have one, or a maximum if it is, of course, a diploid cell, you'll have two copies of each DNA sequence. Now, to actually perform sequencing, you need to have a lot more of the template. So in contrast to previous techniques where PCR amplification was being done prior to any other step, here, after the library is hybridized onto a flow cell, this amplification step is carried out. For now, just take it as it is. We'll understand it using a video in the next class. Now, these amplified clusters are then subjected to sequencing. And for sequencing, here also, what is used is the labeled primer. So during this amplification step, whichever base keeps on getting incorporated gives a readout in the form of fluorescence, emitted fluorescence, and that fluorescence gets recorded, and you will have these on the flow cell. You will see these localized regions where the same base gets incorporated at the same time, and then that can be detected. So basically, what you effectively are doing is very similar to the Sanger method that we just discussed, except of course there chain termination was taking place. Here, instead, what happens? Each base that gets incorporated at each position releases one type of a fluorescent emission. That emission is recorded. The unincorporated nucleotides will obviously be washed away. So, for each position, when amplification is taking place, each of the four bases will try to bind. Whichever base finds complementarity will be encoded and its fluorescence will be detected. Each synthesis is followed by the use of a laser, like I just mentioned during the sequencing step. That is what happens. And the laser eliminates the fluorescent probe and blocking group. The subsequent cycle starts when one of the four bases develops a measurable fluorescence color distinct to it that allows for the sequence to be identified. This will continue until the entire template or DNA molecule is sequenced, and you will have the final readout. So key differences between the previous technique and this technique. First of all, you're using the entire genome. Second, you're using adapter ligation. You're fragmenting the DNA to generate some readable sequences. Fourth, you're preparing a library on a library of all the DNA molecules. Fifth, you are hybridizing it to a flow cell on which amplification is taking place using a specific method, the Illumina bridge amplification method. Then the amplified clusters are actually subjected to DNA sequencing, and the sequencing results are understood by the incorporation of a nucleotide, of the correct nucleotide, and the release of a fluorescence emission, which gets detected every time a base is incorporated. And the final readout is then interpreted based on what fluorescence emission was given out. This will continue for the entire sequence which is being studied. Now, I think I'll keep this for now because this might actually confuse you even further. Let's keep the third generation sequencing for now. This was my last slide. We'll discuss it in the next class. Let's keep it till second generation sequencing. We'll discuss the next one in the next class. If you have any doubts, please let me know. I'll try to clarify it. But of course, I'll come up with more information in the next class. Please let me know. Any doubts, anything you want to be clarified today? Because a lot of diverse things were covered today, and a large part of the class was actually not even audible. So please let me know. Anything you want to be specifically discussed in the next class, then I'll highlight it first. Guys, am I
Ma'am, in
audible?
this class we have, yes, yes, ma'am. Ma'am, in this class we just discussed the sequencing techniques, ma'am.
No. So there was we discussed polymorphisms and their effect. How are they different from mutations? How they are able to influence diseases, and what types of polymorphisms can actually be present. Then the use of functional genomics. What is it and how it can be used? The influence of CRISPR technologies in current scenario for functional genomics, and then we moved on to sequencing technology. So these were the broad topics that were covered. I will
Okay, ma'am.
upload the recorded lectures. Don't worry, I'm waiting for the course portal to open. I've contacted the admin, so they said it'll take a few more days. But if necessary, I'll cover some of this. We can dedicate like half an hour, 40 minutes in the next class to a revision of these topics, and then we'll move forward. And I'll
Ma'am,
discuss
the
NGS
starting
in more
part,
detail. Yeah.
ma'am, like the sequencing techniques are clear to me, but the starting part, the mutation and polymorphism part, actually it is in my
Hmm.
syllabus also, and it is not like
Okay.
it is not very clear to me. These techniques
Okay.
are clear, like Sanger, Maxim Gilbert, these are clear.
Hmm.
But the basic
Okay.
part, like in the next class, can you please elaborate, like before this
Okay. Okay.
slide,
Sure.
in brief,
Sure. Sure. Sure.
just
Yeah. Yeah. No problem. We can discuss it again. I'll
ma'am,
discuss it anyway. Yeah. Yeah. Go ahead.
ma'am, like the timing is 6:30. Can it be from 7 o'clock?
From 7. Okay, no problem. I'll inform. Actually, I think I mailed that the class is from 6 p.m. Was it 6:30 in the mail by
Uh,
chance?
um, that is what I'm saying, ma'am. Actually, I have a meet from 6 to 7. That is why I am like
Okay.
that time I
Okay,
will
you're not
not be
able
able
to
to join.
join.
Yes,
Okay,
ma'am.
okay, no problem.
Is
Yeah, we can
ma'am, if
shift it to
if it's
7.
possible. Yes, ma'am. Yes, ma'am.
Yeah,
Please,
no
ma'am.
problem. We can keep it from 7 to 8:30, 8:45. Basically 9
Yes.
up to 9. We can shift
Okay,
it,
ma'am.
no problem. Okay,
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 thank
okay.
you,
I'll
ma'am.
send an email for the next class and we can discuss this these initial topics before
Yes.
proceeding. Okay,
Thank you, ma'am.
okay, no problem. Anything else you would like to suggest? Otherwise, we'll end the meeting for today.
No, ma'am. From my side, that's it.
Okay, okay. Okay, then thank you so much for joining. I'm very sorry about all the technical glitches today, but we'll make up for it. Don't worry. And I'll share the presentations with you over email because the course portal has been not accessible for over 2 weeks now. So I'll mail them to you. In the meantime, if there's anything else from previous classes or from today's class, please let me know. We'll discuss it. It's a long course, so there's of course, a possibility you will keep forgetting the initial things as we move on ahead. But please let me know. We'll have an interactive session again.
Thank you, ma'am.
Okay.
Thank you,
Thank
ma'am.
you. Have a good night. Thank you. I'll end the class.